The now how we will be culturing our buds. Either we are taking the terminal buds or we are taking the axillary buds. How we are able to culture these buds under in vitro conditions. How these buds are cultured in vitrocally. So here we are taking the terminal buds. These are our meristem, or we call these a shoot tip. We have to isolate these shoot tips. Then these shoot, when this shoot tip is directly taken, or the meristem culture, then from these tips, we are directly developing shoots. Then they are forming the auxiliary branch. And then this, after forming the auxiliary branches, we are transfer this plantlets into the new medium where they will be able to form roots, or they will be able to form roots. In auxiliary bud, what we are doing, we are taking the axial buds. We can directly take the auxiliary bud and transfer on the liquid medium. Then the shoot will develop, or we can take the node. We can take the 2-3 auxiliary buds together. Then we can place it on the in the liquid medium where many shootlets will develop. Then the auxiliary branching will take place. Then again the rooting process will take place. For shoots, we have to give oxygen concentration less, but cytokinin concentration should be more. And for formation of the roots, we have to take growth hormone, oxygen more, and cytokinin less. You already know this thing. Now the another culture is our endosperm culture. Another culture is our endosperm culture. Endosperm culture. What is endosperm? It's a tissue which is present inside most of the seeds in case of flowering plants. जो flowering plants है, जो उनके seeds है, उनमें एक tissue होता है present, जिसको हम लोग बोलते हैं endosperm. So if we will open up the seed, what we will see there? We will see a embryo along with the tissue. That tissue part is our endosperm. Its function is to provide nutrients to the growing embryo. जब हम लोग एक seed को soak करते हैं, किसमें पानी में पहले soak करते हैं. So what is happening to this seed? That in presence of water, sunlight, and the appropriate temperature, the dominant effect of the growth hormone, that is our, I don't remember this time. So dominant effect of the growth hormone, that is abscisic acid, exactly, is getting decreased, but there is the formation of more and more gibberellic acid. This gibberellic acid go into the nucleus where they initiate the transcription of alpha amylase enzyme. Then alpha amylase enzymes are transcribed, then the translation, then the protein are formed, alpha amylases are formed. Then this alpha amylase broke these starch and they make this starch available to the growing embryo. This embryo will develop into plumule and radicle. The endosperm doesn't have only starch present in it, they have ample number of proteins as well as oil. So this is overall, we can say that this endosperm is also good source of nutrient to the humans also. Like the best example is our coconut water. That is actually the endosperm. This endosperm, if we will open up, if we will visualize this endosperm, they don't have well-developed vascular differentiation there. So what do we do? They only provide the nutrients and to the plant. And secondly, they are triploid in nature. In 1930, scientists had tried to go for embryo endosperm culture under in vitro conditions. So depending on the plants, the endosperm may be consumed during seed development. Sometimes this endosperm is utilized, sometimes it's not utilized. It depends on the plant which you have taken. So depending on the plants, the endosperm may be consumed during the seed development. Like we can have different examples, cucurbits, legumes, mulberry are there, or sometimes these endosperm persist in the seeds, or these endosperm persist in the seed as a massive tissue, like palms में है या castor seed जो हमारे हैं उनमें जो हमारा endosperm है वो वहां पे persist करता है. This endosperm culture is having, we can culture this endosperm at a mature state and at a immature state. So when this endosperm is cultured at a mature stage, we call this as a mature endosperm culture. It is the in vitro culture development of an isolated mature endosperm from a ripened endospermic seed. The seed should be ripened. Then this endosperm is placed on a suitable culture medium. From this endosperm, we are able to obtain triploid plants. We are able to obtain triploid plants. And such type of culture is called as mature endosperm culture. The another is immature, as name indicates, immature. It is an isolation of immature endosperm. When the seeds is not fully developed or fully mature, then we are culturing this endosperm, immature endosperm on a suitable medium. Again, we are able to get plantlets from these immature endosperm, and the culture is called as immature endosperm culture. The another culture which we have to discuss is our protoplast culture. Okay, before going to protoplast culture, what is the protocol for endosperm culture? What is the protocol? We are having mainly 3 steps. The immature or mature seeds are dissected. First, we have to dissect how we will be able to isolate or get the endosperm. We will take a mature or immature seed. We will dissect it under aseptic conditions. Endosperm is taken, excised along with the embryo. This excised embryo are cultured on a suitable medium, and embryos are removed after initial stage. जब इसकी induction start हो जाएगी किसकी endosperm की, then afterwards when it has got induced so that this endosperm is ready for the cell division, then we can remove our embryo from it. Callus formation takes place, and this process is called as embryogenesis, or directly shoot bud differentiation takes place. From there, the complete plant is formed. From this complete plant is formed. Now, the last part of this lecture is a protoplast culture. We can call protoplast of a plant cell as a naked plant cell because it doesn't have cell wall, or we can call these as a single system, complete plant system where we are able to grow, where we will be able to grow whole plantlet from it. Protoplast culture is the culture of the entire plant cell without its cellulosic cell wall. So protoplast means the cell without the cell wall. This cell wall can be removed either mechanically or enzymatically. With the help of method instruments, we can remove this cell wall by using specific techniques. Like we are having many examples are there. By heat shock treatment, we are able to remove the cell wall, or by enzymatic treatments, like we can use cellulose, hemicellulose, pectinases to remove our cell wall. Then this all the process which we have to carry in case of plant tissue culture has to be carried aseptically. Then this protoplast is taken and cultured on the suitable medium. Then from this, the whole plant can be developed. Now, what is the importance of this? What is the importance of the protoplast culture? As I said right now, the single cell is it's a complete whole plant. We will be able, it gives us a best experimental system where we are able to study our plant. This will be best where we will be able to study our whole plants, how things are happening, how developmental processes takes place. Even we can form, with the help of this protoplast, we are able to get hybrids. We will take two protoplasts of two different species which are sexually incompatible or physiological. They are not able to mate. Under the in vitro condition, we allow them to mate to form the hybrids. Then these hybrids can be used in crop improvement programs. These can be used in crop improvement programs. Even we know that protoplast, when there is no cell wall, the plasma membrane are having ability to go for endocytosis process. So we can place our gene of interest outside the plasma membrane. Then this gene of interest or any mutagen will be ingested into the plant cell, where they incorporate into their genome. Sometimes we have to go for lots of screening where we will be able to get the hybrid seeds. So we are able to insert our foreign material by the endocytosis process in these protoplast cultures. These when protoplasts are placed in the liquid medium, then slowly and gradually cell wall formation takes place. This protoplast system provides us the information what is the mechanism in the formation of the plant cell wall, and how biosynthesis and the deposition of cell wall takes place under natural conditions. So even we are able to, this is a complete whole cellular system of the plant. We can even go for manipulation. We can go for manipulation as we do manipulations in case of microorganisms. We can place gene of interest outside, or we can, मतलब कोई भी छेड़खानी हम इसके साथ कर सकते हैं. Even these protoplasts can be directly inserted with the foreign gene. Then these protoplasts, we will see how the inserted genes are interacted in the cell with different components of the cell. What is the mechanism, or how the things are taken up by the cell under in vitro conditions. So all this is important to understand the plant tissue culture betterly. So now how we are going to, what is the protocol for the protoplast culture? We will take young leaf, young plant. Young plant is having always more meristematic activity. We will take leaf from it, because in leaf we are having presence of mesophyll cells, and there is intracellular space between the cells. So the space will allow enzymes to break the cell wall easily. So we take epidermis, we peel this epidermal cell, then we fragment this epidermal cell by plasmolysis, and then the cells are treated with enzyme mixtures, either by cellulase, hemicellulase, and pectinases. These enzymes break up their cell wall, and there is the release of the protoplast. So with the help of the centrifugation, mild centrifugation, we will spin it for some time, so that the debris of our plant cell wall will be at the top, and the pellet will be the protoplast. Then the isolated protoplast, we will go isolate it, we will culture it on our solid medium, or the liquid medium is preferable. So we can place it on the Petri plates, where then the regeneration of cell wall takes place. Then these cell form the cell clumps. Then from these cell clumps, callus tissue is formed. Then from the callus tissue, our plantlets are formed. Then these plantlets, which are growing in the tissue culture labs, are placed on the soil, and the process is called as hardening. Then when these plantlets develop or form their roots in the soil, then from the lab culture, these plantlets are taken to the greenhouse. Then when these plantlets develop fully, then from the green plantlet, they are selling in the market. These were all cultures for our plant tissue culture.